conclude, circular RNAs represent a new class of regulatory RNAs which may prove to be future drug targets or biomarkers for the diseases of aging. I'd just like to thank the people that did the work, and in particular, I'd like to draw your attention to this lady here, who is Jacques. She carried out all of the circular RNA work that I presented today. Like any big study, this has there are lots of people who've been involved here. Members of my own team, and in particular, I'd like to thank Jacques. Also collaborations with Luigi Ferrucci and Stefano Bandinelli from the National Institute of Aging in America, who are the PIs of the Chianti study, and Luann Peters and Kevin Flecke from the Jackson Laboratory, who helped us out with the mice. And with that, I'm happy to take questions.
Okay, thank you very much, Dr. Harris, for the presentation. And so I'd like to open the session for questions. Anybody has questions? You can write in the chat box, or else we can directly ask Dr. Harris. Any questions?
I'm putting my email in here in case anybody thinks of anything later.
Yes. Okay, Dr. Harris already shared her email ID, so you can write your questions to that email ID if you have any. Okay. Okay, that's fine. I'd like to thank Dr. Harris for the presentation. So we'll move forward to the next presentation from Dr. Jacques Tremblay from Laval University, Canada. Dr. Jacques, you hear me? Could you please unmute your audio? Oops. I don't hear. Okay, that's fine. Okay, good morning.
Good morning, everyone.
Yeah.
I will try to share my screen. Sorry, I'm not able to share so far.
Okay, do you need any help from our side?
Yeah. I have a MP4 presentation and I would like to share it.
Okay, first of all, you need to open your presentation.
Yeah, it's open.
And then you come to Zoom application.
Yeah, the Zoom application I have also.
And click on the share screen option.
Yeah.
So you can see the windows, all the windows that you have open.
Yeah, that's the problem. I don't see the windows that I would like to share. Okay, I'm clicking on share, and then there's whiteboard, Google Chrome, unknown, QuickTime Player is the one that I would like to share, but it doesn't. Can you cannot see my screen, huh?
Yes, I don't see.
Share screen. When I'm trying to share, it opens a open.
Okay, if it's taking time, then
I think you guys have my MP4 presentation. Can you play it for me?
yes, yeah, I do. Meanwhile, you can try your luck with your that to open it.
The title of my conference. Yeah, that's the one.
Yes, I'm sharing it right now.
Is insertion of the protective Icelandic mutation A673T in the APP gene using the CRISPR-Cas9 base editing technology. The title of my conference is Insertion of the protective Icelandic mutation A673T in the APP gene using the CRISPR-Cas9 base editing technology. On the editing of the amyloid precursor gene. The sound is not playing correctly. Use a Cas9 protease eventually of sporadic Alzheimer. It is based on the editing of the amyloid precursor gene. How come you don't have the sound correctly?
Yes, the voice is missing here.
Technical problem. Alzheimer would be due to the accumulation of beta amyloid peptide derived from the amyloid precursor protein. These peptides form amyloid plaque, leading to hyperphosphorylation of the tau protein, neurofibril accumulation, and neuron death.
So you can take any of anyone's help for that.
Yeah, well, the amyloid precursor protein can be cut by the alpha secretase and the gamma and the beta secretase, can be cut by the alpha secretase and the gamma secretase, and this produce fragments that do not cause any problem. But it can also be cut first by the beta secretase, followed by a cut by the gamma secretase, and this produce fragments of the beta amyloid peptides that do accumulate and form the amyloid plaques. And some people have tried to block the beta secretase, also called BACE1, as a potential treatment for Friedreich attack, for Alzheimer's disease. So currently there is no treatment for Alzheimer. Some people are trying to use monoclonal antibody to try to degrade the beta amyloid peptides and the plaques that are formed by them. Some other people are trying to increase the nerve growth factor. I don't know. That's completely crazy. The slides are not moving at the right time. So our approach is to try to block the degradation, the formation of the beta amyloid peptides by introducing a mutation that will prevent the cutting by the beta circuitase. We are using the CRISPR-Cas9 technology to do that. That technology uses a Cas9 nickase, and a guide RNA which recognizes a sequence of 20 nucleotides. And this permits to introduce a cut at a very precise site in the target gene. I'm very sorry, the sound was pre-recorded and it seems that they are not able to play the sound. When you induce a double strand break like that, you can have a repair by non-homologous enjoining and by homology-directed repair, but both of these approaches are not working very well, and you have to be able to select the cells which are modified. We are instead using the Cytidine-based editing technology, which use a Cas9 nickase, which is fused with the Cytidine deaminase, and this permits to chemically modify the Cytidine into a thymine. And this will reduce the formation of amyloid plaques because the A673T mutation is a normal mutation that has been observed in the Icelandic population, and the people that have this mutation, they are protected from being Alzheimer. Thus, our objective was to introduce this A673T mutation in order to, in the genome containing, very sorry, very sorry. And therefore, we're trying to introduce that mutation in the genome in order to prevent the development of Alzheimer's disease. Of the protective effect of the Icelandic polymorphism. We have tried to introduce that mutation in cells. In fact, we have created plasmid that contained either a familial Alzheimer's disease mutation or contained the familial Alzheimer's disease mutation and the A673T mutation. And we observe that when the A673T mutation is present at the same time as the familial Alzheimer's disease mutation, we have a reduction of the production of A-beta 40 peptide and a reduction of A-beta 42 peptide for many of the familial Alzheimer's disease mutation. So as you can see, for most of the familial Alzheimer's disease mutation.
You want me to go back to the last two slides? Can you explain that?
No, but can you play the sound that was already pre-recorded? That would be so convenient.
Let me try it once again, please. Hold on a second.
Very sorry for everybody. You know, this was a pre-recorded MP4. I'm not able to share my screen, and it seems that they are not able to play correctly my MP4 movie. of my conference is Insertion, Preventive Treatment of Familial Alzheimer's Disease, responsible for 70% of
I think now I'm succeeding. I'm sorry for that.
affects about 50 million people around the world. There are two forms of sporadic or late onset, which is 98% of the Alzheimer's cases that affect people which are more than 65 years old. And there is familial or early onset disease, which are responsible for about 5 to 10% of the cases. According to the amyloid cascade theory, Alzheimer would be due to the accumulation of beta amyloid peptide.
We don't see the presentation. We can hear the sound, but we don't see the presentation.
Leading to hyperphosphorylation of the tau protein, neural fibril accumulation, and neuron death. The amyloid precursor protein is cut by several secretases, alpha, beta, and gamma secretases. The cutting of the amyloid precursor protein by the alpha secretase followed by a cut by the gamma secretase produce protein fragments that are degraded without causing any problem. However, APP may also be cut first by the beta secretase, followed by a cut by the gamma secretase, and this produces beta amyloid peptides, which are 40 to 42 amino acid long, which accumulate and form the amyloid plaques. The Icelandic A673T mutation was discovered in 2012 by Johnson et al. This mutation prevents the development of Alzheimer's disease, even in people that are more than 95 years old. It changed the cutting site of the beta secretase to a position which is at 11 amino acid further. There is currently no treatment for Alzheimer's disease. There are some indirect approach that people are trying to use monoclonal antibody against the beta peptide to increase their degradation, or trying to increase the nerve growth factor to improve the survivals of neuron. And there is also some direct approach, which is the prevention of plaque formation using inhibition of the beta secretase, also called BACE1 enzyme. However, inhibition of that enzyme produces adverse effects. I support that the definitive treatment of Alzheimer's disease is gene therapy. The original CRISPR-Cas9 technology used a Cas9 nuclease in yellow here and a guide RNA, which has a part of 20 nucleotide which recognizes one of the strand of DNA and a constant sequence in red. This Cas9 nuclease forms a complex, therefore, with the guide RNA and with the DNA, and induce a double-strand break at exactly 3 nucleotides from the PAM, which is NJJ, for the Cas9 of Streptococcus pyogenes. The double-strand break, which is induced by the Cas9 nuclease, is repaired either
Dr. Tremblay,
Yeah.
We can't.
I think I succeeded it. I'll play it from the.
Yeah, we are hearing the sound, but we're not seeing the presentation.
title of my conference is
We can't see the PowerPoint.
Yeah.
Icelandic mutation A673T in the APP gene. Beta amyloid form
So I'm just playing from this slide.
Icelandic A673
Yeah, now we have the slides.
peptides, which are 40 to 42 amino acid long, which accumulate and form the amyloid plaques. The Icelandic A673T mutation was discovered in 2012 by Johnson et al. This mutation prevents the development of Alzheimer's disease, even in people that are more than 95 years old. It changed the cutting site of the beta secretase to a position which is at 11 amino acid further. There is currently no treatment
for Alzheimer's disease. There are some indirect approach that people are trying to use monoclonal antibody against the beta peptide to increase their degradation, or trying to increase the nerve growth factor to improve the survivals of neurons. And there is also some direct approach, which is the prevention of plaque formation using inhibition of the beta secretase, also called BACE1 enzyme. However, the inhibition of that enzyme produces adverse effects. I support that the definitive treatment of Alzheimer's disease is gene therapy. The original CRISPR-Cas9 technology used a Cas9 nuclease, in yellow here, and a guide RNA, which has a part of 20 nucleotide which recognizes one of the strand of DNA, and a constant sequence in red. This Cas9 nuclease forms a complex, therefore, with the guide RNA and with the DNA, and induce a double-strand break at exactly 3 nucleotides from the PAM, which is NJJ, for the Cas9 of Streptococcus pyogenes. The double-strand break, which is induced by the Cas9 nuclease, is repaired either by non-homologous conjoining or by homology-directed repair. However, this is not the technique that we are going to use today. We are going to use instead the Cytidine-based editing technology. It's used in Cas9 decades, which cuts only in one little strand of DNA. And this Cas9 is fused with a Cytidine deaminase, which permit to chemically change Cytidine into a thymine. Our hypothesis is that the accumulation of amyloid plaque may be reduced by inserting the Icelandic natural variant A673T with the base editing technology. Our project had 2 objectives. One, to demonstrate the protective effect of the Icelandic mutation A673T in presence of another familial Alzheimer's disease mutation. And 2, introduce the Icelandic mutation in vitro with the CRISPR-Cas9 base editing technology in 293 T cells in human neuroblastoma and in neurons. Let's first introduce the demonstration of the protective effect of the Icelandic polymorphism. This was done by transfecting neuroblastoma with plasmid coding for the amyloid precursor protein gene with one or several familial Alzheimer's disease mutations. The plasmid transfected in the neuroblastoma contained only the APP gene with one familial Alzheimer's disease, or contained not only the familial Alzheimer's disease, but also, in addition, the Icelandic mutation. The presence of the Icelandic mutation in the plasmid coding for APP with one familial Alzheimer's disease mutation led to a reduction of the production of the A-beta-40 peptides. The presence of the Icelandic mutation, in addition to the familial Alzheimer's disease mutation, also led to a reduction of the A-beta-42 peptides. For 57% of the familial Alzheimer's disease mutation, the addition of the Icelandic mutation reduced by at least 20% the production of A-beta-carote and A-beta-42 peptides. The London mutation is one of the most frequent familial Alzheimer's disease mutations. The addition of the Icelandic mutation to a plasmid coding for the APP gene with the London mutation reduced by 80% production of A-beta-40 peptides and by 62% the production of A-beta-42 peptides. In our second experiment, instead of using plasmid already containing the Icelandic mutation, we aim to introduce that mutation in vitro cells by using the Cytidine-based editing technology. We have tested several different cytidine deaminase enzymes. The cytidine deaminase can convert cytidine into thymine in a nucleotide window of about 15 nucleotides from the photospacer adjacent material, which is NJNJ for the SP-Cas9 PDR decades variant that we have used. This permits to convert the alanine codon. In fact, we are converting the antisense codon, which contains C, and transforming that into a TGT antisense codon, which is the antisense codon for the threonine. This is a list of the 14 different cytidine-based editors that we have tested in order to identify the best one to introduce the Icelandic mutation in the APP gene. Plasmid coding for these various cytidine-based editors were transfected in neuroblastoma. The DNA was extracted 72 hours later, and deep sequencing was used to quantify the percentage of cytidine that were modified into a thymine, thus introducing the A673T mutation. The positioning in the genome of the Cas9 decase, which is part of the cytidine deaminase enzyme, requires the presence of a single guide RNA that detects a sequence of 20 nucleotides. We found that the presence of 2 single guide RNA in the plasmid coding for the SP-Cas9 decase fused with the cytidine deaminase gave better results. Among the various cytidine-based editing enzymes that we have tested, the target A deaminase coupled with the SP-Cas9 decase VQR gave the best results. Plasmid codings for the target 8 cytidine deaminase fused with the Cas9 decase VQR variant were transfected into neuroblastoma containing either the APP wild-type gene or the APP gene with the London mutation, and the results of deamination were quantified by deep sequencing of the APP gene, and the A-beta and A-beta-42 peptides were quantified by the ELISA technique. The insertion of the Icelandic mutation by deamination of the cytidine by the base editing technique reduces the secretion of A-beta-40 and A-beta-42 peptide by neuroblastoma containing either the APP wild-type gene or the APP gene containing the London mutation. Following the insertion of the Icelandic mutation in neuroblastoma cell line, we also wanted to introduce this Icelandic mutation in real neurons. We have differentiated into neurons fibroblasts obtained from 2 different mouse models of Alzheimer's disease, and fibroblasts obtained from the patient with the familial form of Alzheimer's disease, that is the London mutation. The differentiation of fibroblasts into neurons was obtained by infecting the cells with a lentivirus coding for 2 different transcription factors, inducing the differentiation of fibroblasts into neurons. Neurons were also obtained by culture of hippocampal neurons obtained from prenatal mouse. This is an illustration of the differentiation of fibroblasts in neurons. The immunofluorescent staining of the tau protein in D confirmed the differentiation of the fibroblasts into neurons. This is an illustration of a neuron culture derived from an embryonic mouse hippocampus. Again, the immunofluorescent labeling of the tau protein in green confirmed that the presence of Infections of neurons with a lentivirus coding for target A SPCat9PK permitted to deaminase the targeted Cytidine to convert the alanine to a threonine, thus inserting the A670 mutation in neurons. Infections of neurons either from mouse origin or human origin with a dual APP virus or with lentivirus coding for target 8 SPCat9PK permitted to deaminase the target Cytidine, thus introducing the A673D Icelandic mutation. This reduces the secretion of A-beta-42 peptide. There are still many problems to be resolved before this approach of introducing the Icelandic mutation can be a treatment for familial Alzheimer's disease and for sporadic Alzheimer's disease. We need to improve the transduction of neurons. The dual AAV system has to be improved, and we have to optimize these vectors by optimizing the promoter and the Cytidine deaminase Cas9 complex. One of the problems of using the Cytidine-based editing technique is that it modifies several Cytidine which are located within a 5 to 6 nucleotide window. Thus, when we are using the nucleotide C2 to introduce the A673T mutation, we are also modifying to a lesser extent the C1 nucleotide, thus modifying another amino acid and introducing the E674K mutation. So far, our results suggest that this E674K mutation also contributes to reduce the cutting by the beta secretaries. However, this mutation may also affect other characteristics of the APP protein or of the A-beta peptide. Will that affect aggregation? We don't know. We have just recently switched to using the prime editing technology instead of using the Cytidine-based editing technology. This prime editing technology was developed by Laval in 2019. It used a Cas9PKs fused with a reverse transcriptase, and it used a modified single guide RNA, which is called a PEG RNA. This PEG RNA, as a single guide RNA, has a 20 nucleotide sequence which interacts with one strand of DNA. Then it has a constant part of the single guide RNA, and two sequences are added to the single guide RNA. One sequence is a primer binding site, which is 12 to 15 nucleotides in length. It permits to interact with the free strand of DNA. And then there is a reverse transcriptase template, which specifies what DNA strand has to be synthesized by the reverse transcriptase. This reverse transcriptase template may contain one or several nucleotides which are modified and will indicate what mutation has to be introduced in the new DNA strand synthesized by the reverse transcriptase. The great advantage of the prime editing technology compared to the Cytidine-based editing technology is that this is a more precise nucleotide editing technique. It permits to modify only the C2 nucleotide without modifying the C1 nucleotide. The prime editing technology can be repeated several times on the same cell pool. We have done that for the Emx1 gene and for the APP gene. In fact, we have done 6 repeated treatment with the prime editing technology. And this permitted us to introduce a nucleotide mutation in 79% of the Emx1 gene and in 59% of the APP gene. prime editing technology made us be a better method than the Cytidine-based editing technology to insert the protective Icelandic mutation. However, so far we have introduced the Icelandic mutation in cells and culture. This mutation will have to be introduced in neurons in people. We have nevertheless been able to reach several conclusions. The Icelandic mutation A678T has been shown to reduce the secretion of toxic amyloid peptides in vitro, even in the presence of some familial Alzheimer's disease mutation. We have successfully inserted the Icelandic mutation by base editing in up to 57% of the APP gene, and with the prime editing technology in up to 59% of the APP gene. Cas9 expression was observed in neurons in vitro following infection with antivirus, and we need to optimize the neuron transduction for the in vivo experiments. Thank you for your attention.
Thank you very much for the audience. I'm sorry for the problems during the presentation. You know, it's the first time I do a pre-recording of my conference, and therefore I did not practice the conference because I was confident that everything was pre-recorded, but that played a big trick on me. The recording was, the sound was, I don't know, the sound was good for you.